the two membrane, because mitochondria is a double membranous cell organelle. So there is a space between outer mitochondrial membrane and inner mitochondrial membrane. So in between the space between two membrane, that is perichondrial space, possibly this reaction is happened in which pyruvic acid is decarboxylated. Decarboxylation simply means removal of carbon dioxide. So if from a 3-carbon compound, that is pyruvic acid, if we remove one carbon dioxide, then the molecule, the compound will form this acetyl coenzyme A, which is just a 2-carbon compound. So in this reaction, if we simplify the reaction, pyruvic acid, which is a 3-carbon compound, as per the name, decarboxylation, one carbon dioxide is released. So from 3-carbon, if one carbon dioxide is released, we have 2-carbon compound, that is acetyl coenzyme A, which is a 2-carbon compound. And during the reaction, we have one molecule of NADH2 formed. It is also serve as source of energy, just like the NADH2 formed during glycolysis. So gateway reaction is a simple reaction that link the mitochondria from cytoplasm, because pyruvic acid is formed in cytoplasm and rest of the reaction occurs in mitochondrial matrix. So during the transport of this pyruvic acid into the mitochondria, this reaction is completed and one carbon dioxide is released and the compound form will be 2-carbon compound, that is simply acetyl coenzyme A. So from this 2-carbon compound, because the final product during respiration, during aerobic respiration, as I have said earlier, because these reactions can occur only in presence of oxygen. So if there is aerobic respiration, then second, third and fourth reaction will happen. So as the final product from the oxidation of glucose is carbon dioxide and water, and we have still a 2-carbon compound, that is acetyl coenzyme A, it means that 2 more carbon dioxide is also released from this compound. So the third step is simply known as Krebs cycle. After the discoverer, he is a Nobel laureate and received Nobel Prize for this work. It is also known as, because we have oxidation of this compound, so it is known as oxidative decarboxylation. So it's Krebs cycle or oxidative decarboxylation. It is also known as TCA cycle, that is tricarboxylic acid cycle or citric acid cycle, as the first stable product form will be citric acid. So it is also known as TCA cycle or citric acid cycle that we discuss in the detail Krebs cycle. But as far as simplifying the mechanism of respiration, third one is the Krebs cycle or oxidative decarboxylation, where this acetyl coenzyme A will, along with a intermediate compound, it will change, it's a 2-carbon compound, it will change into 2 molecules of carbon dioxide, and this intermediate compound is formed as such. This will be same. So in presence of this intermediate compound, which is a 4-carbon compound, it combines with 2-carbon compound. First product will be the 6-carbon compound, and during the complete cycle, we have again regenerate this intermediate 4-carbon compound, and from this 2-carbon acetyl coenzyme A, we have 2 carbon dioxide molecules released. So from one pyruvic acid, 1 carbon dioxide is here and 2 carbon dioxide is here. It is a 3-carbon compound. So finally, if both the reactions are clumped together, finally we have 1 pyruvic acid molecule which is oxidized into 3-carbon compound, so that they are released. And here is 6-carbon compound have 2 pyruvic acid. So from 2 pyruvic acid we have 6 CO2. So from 1 molecule of glucose, 6 CO2 will be formed. If we simplify the reaction, then glucose equals to 6 O2 and we have 6 CO2 plus 6 water molecule formed. So, but along with this, the maximum amount of energy as per the 4 process released or formed that will be in the form of whether direct ATP, GTP, NADH2, FADH2. So in terms of maximum number of molecules, so these are formed during Krebs cycle. So maximum amount of energy is released by this step, that is the Krebs cycle or oxidative decarboxylation. If we simplify the reaction, then this one molecule of acetyl coenzyme A has 3 molecule of NADH2, 1 molecule of FADH2 and 1 molecule of ATP, that is basically GTP, but we can simplify it as ATP. So from 1 glucose molecule, if we summarize, 2 NADH2 are here, 1 NADH2 is here, 3 NADH2, 1 FADH2, 1 ATP and GTP and so on. As far as ATP direct from during the process, there is a term used called substrate level phosphorylation, because that substrate is any compound that depends on the reaction. So substrate level phosphorylation means phosphorus is added to ADP. That phosphorus which is added to ADP comes from the substrate, maybe anyway, and we have formed ATP. So if ATP are formed directly during the respiration, then it is called simply substrate level phosphorylation. There is also the carrier of the energy in the form of FADH2 and NADH2. So if FADH2 or NADH2 is formed, it is called oxidative phosphorylation. That completes in the 4th step. One thing which is most important in this case, directly 36 or 30 ATP are not formed during these 3 steps. Mostly the energy is in the form of NADH2 or FADH2. These are the carrier of energy. So in the 4th step, this NADH2 or FADH2 is oxidized and then finally ATP will yield. So that comes for the 4th. So it is simply called electron transport system. Or ETS. So ETS is the 4th step. As far as ATP is formed by the oxidation, it is also known as oxidative phosphorylation. So 4th step is the ETS or oxidative phosphorylation that occurs in inner mitochondrial membrane. Or Christie. So that occurs in inner mitochondrial membrane or the membrane of Christie. These are the infoldings which are formed in the inner mitochondrial membrane, and these infoldings are known as Christie. आपने mitochondria का structure पढ़ा होगा, उसमें ये Christie term use करते हैं. So we can use both the terms, either inner mitochondrial membrane or Christie. So ETS occurs in inner mitochondrial membrane or Christie. And the purpose of ETS is the conversion of NADH2 into 3 ATP or FADH2 into 2 ATP. So the FADH2 or NADH2 which are formed during these 3 steps, 1st, 2nd and 3rd, from 1 glucose we have 2 NADH2 in glycolysis. From 1 pyruvic acid, we have 1 NADH2 in the gateway or link reaction, that is pyruvate decarboxylation. And in the 3rd step, that is Krebs cycle, we have 3 NADH2, 1 FADH2. And these NADH2 or FADH2 yield 3 or 2 ATP. It is shown here. NADH2 will yield 3 and FADH2 will yield 2 ATP during ETS. So the maximum energy as far as the molecule are formed here, but they will be only the energy carrier. Energy in the form of ATP is obtained only when ETS is completed in inner mitochondrial membrane. So if ETS is not happening, then there is no ATP or NADH2 will form during NADH2 or FADH2. So the 4th step is the ETS, that is simply the oxidative phosphorylation. So if overall summarize the complete chapter, we can say, because in the whole chapter we have discussed these detailed process one by one step. That one is the glycolysis, simply the pyruvate decarboxylation, Krebs cycle, and last one is the ETS. And if we overall summarize the reaction, we have 36 or 38. What is the difference between 36, 38? That will be discussed later, because in some organism the calculation will be 36 or in some the calculation will be 38. But if we summarize, then from one glucose molecule we have 36 or 38 ATP formed during aerobic respiration. That is the most important thing, because we have condition here. The first reaction, that is glycolysis, is common for both aerobic as well as anaerobic, but the second, third and fourth reaction will happen only in presence of oxygen, and so that we have finally from one glucose molecule 36 or 38 ATP formed during aerobic respiration. Clear? So if we sum up these reaction, it will be written as